The other division of the adaptive immune response involves B cells. And B cells work in kind of slightly differently than T cells, although there is some overlap between the two systems. So first of all, B cells do not require antigen presenting cells like T cells do. So they can actually identify antigens on pathogens directly. However, they do get activated by helper T cells. So the helper T cell is kind of this intermediate that not only activates the killer T cells, but it also activates the B lymphocytes as well. Once the B cell is activated, it is cloned and it either develops into plasma cells or memory cells. So we'll take a look at the plasma cells first. The plasma cells are specialized cells that produce antibodies. And antibodies are these little proteins that are able to move in the body fluids like the bloodstream and the tissue fluid, and they attach to bacteria. So they attach to bacteria, they attach to the antigens, and they basically mark them for destruction. So things like phagocytes come along and they see these bacteria with these antibodies attached to it, and it helps the phagocyte know that this is a cell that it should attack and break down. And just like T cells, the B cells also form memory cells. So the next time that exact same pathogen enters the body, the memory cells are activated much more quickly, and the second time around you typically don't know that you've even been infected because you don't develop any symptoms. So that was just kind of a little cartoony overview of the B lymphocytes, but we'll take a little closer look at some of those different structures there. So as we mentioned, the B cells are directly activated by antigens, so you don't need antigen presenting cells in order to activate them. Once they're activated, although we did say that the helper T cells do help with the activation, but they don't require the helper T cells in order to be activated. Once the B cells are activated, they differentiate into the plasma cells, which secrete antibodies, and then the memory cells, which are able to respond the next time you're exposed to the same pathogen. Now, antibodies are also called immunoglobulins or gamma globulins, and an antibody is something that is able to bind to a specific antigen. So antibodies and antigens fit together like puzzle pieces or like a key in a lock. And the word antigen actually comes from antibody generating because it's a molecule that is able to generate the production of antibodies. There are 5 different classes of antibodies, and you can remember them because I spell out the word MAG. So the the IG stands for immunoglobulin, and then M is one type of immunoglobulin, and then there's IgA, IgD, IgG, and IgE. And each one corresponds to a different type of pathogen. So one type of antibody might be present when bacteria is the pathogen. Another one might be more present when a parasite is the pathogen. And another one might be involved in the allergic response to pollen. We're not going to go into the detail of which ones respond to different pathogens, but doctors can actually do blood work and look at which immunoglobulins are present in the blood, and that helps give them a clue as to what is causing the illness. So antibodies actually have several different functions to them. So first of all, they cause clumping. And this clumping is called agglutination. And we talked about this a little bit when we talked about blood typing. But you can see here that the antibodies are the little Y-shaped molecules here, and they're helping to clump together the pathogens. This makes it easier for a phagocytic cell to come along and eat it. So imagine a phagocytic cell trying to find just a single bacterium floating around in the body fluids versus a big clump of them. It's just easier to find. It also helps to label the pathogens for destruction so that the macrophages know that this is something bad that needs to be broken down. It can also activate other immune structures such as the complement proteins. Those were the ones involved in the membrane attack complex that makes water rush into the cell and explode. So it could cause a bacteria cell if the complement were activated. Water could rush into the bacteria cell and cause it to explode. It also just helps to neutralize bacteria and viruses by binding to the antigens, because the antigens of the bacteria and viruses is what helps them bind to cells to infect them. And so the antibodies block those antigens so that they can no longer dock with a target cell and cause an illness to occur or infect that cell. As we mentioned with T cells, the primary response is relatively slow because it takes time for the B cells to be activated and to be cloned and make antibodies. And so the first time you're exposed to a pathogen, you're going to experience symptoms associated with being sick with that pathogen. But after the initial response, those memory cells form. So the second time you're exposed, the response time is much more quick, and those antibodies are released a lot quicker, and the pathogen is disabled before it can cause the symptoms associated with the infection. Now, there are a couple of different types of immunity. And they're actually a combination of either active or passive and natural versus artificial. So let's take a look at what these terms mean, and then we'll kind of talk about them a little bit more in context on the next slide. So first of all, immunity can be active, meaning that someone gets infected with a pathogen and that person's body produces the immune response to that pathogen. So for example, if I get the flu, my body is going to produce antibodies against the flu. That is active immunity. Passive immunity is where immune components can be transferred from one individual to another. So potentially antibodies could be injected in somebody, and those antibodies were maybe produced by someone else and then injected into someone that was sick with the same pathogen. Now, natural versus artificial immunity basically comes with is someone's body creating the immunity, or is modern medicine involved? So natural immunity is something that your body develops the immunity itself, and artificial immunity requires some sort of modern medicine technology in order to occur. So let's take a look at the interaction between these different types of immunity. And first of all, we'll take a look at naturally acquired active immunity. So naturally acquired means that someone, a living thing, produced it. And active means that the person that was infected creates their own immunity. So this is what happens most naturally. So this would be someone being exposed to the flu, they get the flu infection, and their body creates antibodies against the flu, and then they have immunity from that. Now, antibodies can also be naturally acquired, but made by somebody else. And the best example here is antibodies that can be passed from a mother to the fetus through the placenta. So if a woman's pregnant, her baby is protected from the same pathogens that she is, because the antibodies that her body is making get passed to the fetus. Also, when a woman is breastfeeding her baby, antibodies that the mother produces are secreted in the breast milk. And so when the baby is drinking the breast milk, it's getting a naturally acquired passive immunity to that pathogen. And so this is not an immunity that's going to stick around for the rest of the baby's life, but it can keep the baby healthy for the time being. Now, thanks to modern medicine, we also have artificially acquired immunities. So an artificially acquired active immunity is something like a vaccine. So, a vaccine is a situation where they take either a dead or weakened pathogen and they inject it into somebody, and then that person's immune system is activated to develop those memory cells against the pathogen. And the dead or weakened virus is designed to not cause the primary or not cause the symptoms associated with a primary infection, but you get the benefit of that memory or secondary response to that same pathogen. So a vaccine is very similar to naturally acquired active immunity, except for it's artificially acquired, but it's still your body making memory cells against a pathogen. It's just that you're being exposed to that pathogen artificially rather than naturally. And then another way that someone can be kind of given immunity to something is artificially acquired passive. And this isn't very common, but it can occur when perhaps somebody is infected with a pathogen and that person develops antibodies against that pathogen. And then they can actually take the antibodies from the person that developed them and inject them into someone else who has been exposed to that same pathogen. And this was used, kind of the most recent example was with the Ebola outbreak in Africa a couple years ago. So there were some people that, even though Ebola has a very, very high death rate associated with it, there were some people able to develop immunity against Ebola. And with those people, they were able to take samples of their blood and basically extract the antibodies and then inject those antibodies that had been exposed to the Ebola virus. And I don't know how much of a positive effect that had, but it probably helped at least some people not die from it, or at least not have as serious of an infection as they would have.